with the sequence comparison slides and talk about just basic sequence comparison. Now, for a lot of you, this might be very something you know about, very mundane, but I think it's important to go through basic sequence comparison because it's a kind of there's some ideas here that we use in many other places in bioinformatics. It's also a classical problem that was one of the first problems that people solved when they were really dealing with biological data. And I think also, there's kind of this aspect of a lot of times where a classical problem is kind of solved, people recast other problems into this classical mode and solve them that way. In particular, we'll come back to this theme in the class often of dynamic programming and how we can use that to solve something. And many other problems in bioinformatics are solved that way. And a lot of times you can get very good intuition about what's going on by thinking about what people are doing in just very simple sequence comparison and how that would scale up to some other context. And if people have a kind of very physical background, I would very much describe sequence comparison as the simple harmonic oscillator of bioinformatics, if that makes any sense. This is it's a kind of analogy from kind of physics where this is a particular type of calculation that is fairly simple in physics. It's just the simple spring, but really understanding that is really, really useful for understanding a lot of stuff people are doing in physical systems. In any case, that's why, and so much so that people have almost coined that phrase. Okay, so what are we going to talk about? So we're going to talk about doing some basic alignment with dynamic programming. We're going to talk about just a bunch of bells and whistles on that, how we think about suboptimal alignment, how we end gap penalties, how we think of similarity matrices, local alignment, and so forth. So let's just go over the basic problem here. Again, you probably all know this, but I'll just kind of sort of go through this quickly. The raw data you're given is 2 strings, which now obviously these are in real life, they would be much, much longer than the 2 little strings you have here, but for illustrative purposes, we'll make them very short. And you basically want to align them, and then you want to compute some score for how good they align. And if you these obviously are short enough that we can just do this by hand, and we can look at how they match up, and we can see how we can match them this way. They have 2 matches with no gaps. But one of the key things you realize is you can introduce a gap in them and it matches much better. So if you make one insertion, you can match 4 times, but you can actually do that in 2 different ways. You can make the insertion, you see this, because you have 2 T's here, you can match this T in either spot. And then maybe you don't like the idea of making an insertion. Another thing you can do is you can just slide the sequences relative to each other and make a kind of gap at the end. That's another thing you could do. And then you can't get 4 matches, but you can get 3 matches. And so a lot of what you're doing in sequence comparison is you're kind of trying to maximize the number of matches by trading off different kind of, I should say, imperfections, insertions, deletions, end gaps, whatever, in the sequence. So, oops, let me just. So now, of course, if we want to do this for a bigger string, here's an example of it. It's much harder to do this manually. And I think this is a long time ago now. This is, I guess, my God, almost 50 years ago, people started thinking about this. And the people who first started thinking about this were called Needleman and Wunsch, and they provided a classic solution. They didn't really know about the name dynamic programming or a lot of the terms that we're using, but they just kind of came up with a solution. I'm going to kind of literally go through their paper in the kind of classic paper way and show you what they did, because I think it gives you some good intuition of how to solve this. And actually, believe it or not, they were actually doing this alignment before people had even come to standardize on the characters that you use for the different amino acids. So for instance, they have a J in their sequence, and that was actually tyrosine, so be it. So how do they go about doing this? Basically, they took two sequences. Here's one, here's another one, and they sort of lined them up, and they made this kind of square. People often use the term dot plot or similarity matrix for this type of thing. And then they just simply put a tick or a one every place you would get a match. And of course, along the diagonal here, you can see this is what you'd get from matching the sequences up, just straight along. You'd get a certain number of matches, 1, 2, what's that, 3, 4, and 5, and so forth. But you can also match things off the diagonal. So for instance, this C can match here, it could match here, it can match here if you introduce enough gaps. And so this kind of produces this puzzle. And what you want to do is you want to find a path through this puzzle that kind of maximizes things. And actually, you can see this kind of well if I substitute their string with a just kind of fun, sort of fun string here. Sequence analysis is fun, fun sequences are fun. And so you can see how, for instance, this word fun matches a number of places. And what we want to do is kind of match up this matching of sequences to maybe pair it up with another little diagonal that we get from that. So how do they imagine doing this automatically? So I'm going to use their very much their terminology, and like I said, nowadays we might do it a little bit, phrase a little bit differently. But let's just think about how they would do it. They basically kind of went through this thing row by row, and they got to a given point, and they said, "Oh well, what's the best match I could get up to that point?" So if you kind of sort of visualize them going through here, they can match this P with a P, that will be here or something like that. And then they sort of fill in the best value they can get up to that point. So when they come to this square over here where it matches the B with the R, they're not going to get anything from matching that, but they could have added it on to matching the P with the P. So actually get my little, this thing out of the way. They would get a 1 there. So they would fill in the best match up to that point would be a 1. And then they would keep going along. And you can see, for instance, let's see where they would get additional things for that. You could also imagine making a gap. For instance, if you gapped by 1, you could still maintain your match here with the P, and you would also say line up with the B with the L and still get a value of 1. So you can see the 1 keeps going down here, but actually down over here when it was B with B, you would get a 1 here, and you would get the 1 here if you made this huge long gap, right? So you have a 2 total. And then up here, you can see we would have the same situation. We would have, we match the R with the R, and if we had made a little bit of a gap there, we could get both of those in. So then in case that they would start filling this matrix up, and we've kind of talked about the little things that you would match, and you'd keep on going. And you would just keep going through the matrix, filling it up, adding on the match at a given point to the best match you could have up to that point. So for instance, when we're way up here in the matrix, notice that we would be adding on, for instance, let me just see to myself, we'd actually get a 5 over here by adding on to this 4 if we made a little bit of a gap. To do that, because we'd get a 1 from matching the R to the R, okay? And let's keep going. And then we could fill up the whole matrix, and you can see that here, getting all of these things filled in. And then the square that you get at the end, if you fill it up, of course, that's the maximum number of matches you could get. And that represents the best score you can have in this matrix, which happens to be 8 matches by introducing some gaps. Okay. So next thing you might want to do here is trace back. So you'd have to kind of trace back on your path. And so you'd say, okay, I know my best score is 8. Where did I have to come from to get that 8? Well, you would have come from the 7. So that would match the Y with the B, okay, right? Which is a mismatch. Then where would you come from there? Well, you would also come from this 7, which is where you match the C to the C. But notice that at this point here, when you went to the next best match, you could actually do it in a number of different ways. You could match, let me see now, this Y with this Y and skip on that particular thing. Or you could match this N with the N, but you couldn't match the Y with the Y at that point. So there's 2 different possible tracebacks you could do there. And that would correspond here to matching these things, either matching up the Y or matching up the N. And then you would follow your way all the way down, getting all the way down to matching the P with the P. It's kind of, I put Hansel and Gretel here because it's kind of like following your breadcrumbs back, and so forth. And then we just talked about how there's alternate tracebacks in the matrix, and that's highlighted here. And then in addition, you could imagine also not connecting things up on the best path. I mean, you could potentially jump from the 7. You could create alignment that would jump to the 4. Now, obviously, if you did that, your path would no longer be optimum. So you could see what would happen if you followed the 4. If you went from the 7 to the 4, you would no longer have a 7 anymore here because you're not adding it on. You would only get the benefit of the 4. But you could make an alignment, and this would be described as a suboptimal alignment, one that's not one of the alternate optimal alignments, but one that you can still compute fairly easily what value you'd get. So now I want to talk about sort of adding some bells and whistles on. And what I think I'll do is I'll describe all these added things on, then we'll pause at the end and we can kind of think and talk about the whole thing. If you have questions, I'll pause. In terms of adding bells and whistles, there's a bunch of things we can do. So we previously just had the notion of kind of adding in a gap, and we didn't have to pay anything for adding in a gap and so forth. But now what we can do is say, well, we don't want to create a gap in the sequence, and this would obviously correspond to the physical reality of if you're matching 2 sequences, if you have to have a big insertion and deletion, that's kind of unfavorable. So you can introduce a gap penalty, and then the question is, what type of penalty do you want to introduce? You want to say, if I make a gap, for instance, like I'm having here, is that gap a uniform gap? I just pay like a fixed cost, or as I make the gap bigger, do I make it, is it become progressively unfavorable? And people usually describe this gap penalty in terms of a fraction of the overall match score. So for instance, if you get a 1 for matching things, a gap of 0.5 would be like half your match score. And then you could also describe this addition process with actually it's a slope number, like how much additional penalty would you say for making it longer? The way this is written here, you would have a slope of maybe 1/10 of a match for each additional thing you would add on, and so forth. And people often use the term affine penalty for this extension. So now let's do the thing again. And I should point out that I've written a kind of little pseudocode up here for describing the compute that you're doing. And one thing that's neat is you can do this. This is actually written in it's almost like a Excel pseudocode. So you could actually do this whole thing in a spreadsheet. You don't even need to program. You could do the whole thing pretty much in just a simple spreadsheet. So let's see how you would do how this works now. So now you come down here, you get the 1 for the P to the P, you come down. Now you're going to come and try to match what you would match straight along here. You're not making a gap, but now you would come up here when you've kind of come to this one, and you want to match the R with the R. Okay, so now you see, now you know that you can match the R with the R and the P to the P, but you have to make that gap of 1. So here you're going to figure out what's the best you can do to add on in the previous block with the addition of the gap. So for instance, if the gap was a half, you would subtract a half from 2 to get 1.5. Okay, so and now the key aspect of this is you're only looking back all the time. You don't have to look back at the whole matrix. You're just looking back at this kind of shaded black area here, where you're saying, what's the best match I could have up to that point? And by just creating a simple gap. And if you made the, you make a gap of, say, 1 to here, but obviously as we go further, that potential you could consider longer gaps. And so I'm showing all the steps here with the thing in just this simple kind of former thing. This is kind of a useful little slide to see. And now let me explain a little bit of this in slightly more generalized term. So I've been kind of describing how you would do this if you were, you just literally had this little square and you were kind of doing it in Excel or something. But of course, you can think of it as a slightly more generalized idea. And after people, after 1970, when they came up with the solution, people started to think about these type of problems, not this specific problem, but just in general, in a sort of more generalized idea. And this is this idea of dynamic programming. And the basic idea in dynamic programming is to take the best match, or not the best match, the best to solve a subproblem, and then add on to that subproblem, okay? And in the phrase of alignment, you're sort of getting the best alignment you have up to a point, and then you're adding on to that alignment to align the next pair, and then that's now the best you can get up to that point. So here you have imagined you've solved this problem. You're adding on what you can get here, and so you realize, oh, if I make the R of the K and I gap by 1 here, I'll add this on optimally. That becomes the new solved subproblem, and then you're going to add on progressively. And this general idea of solving a subproblem and then adding on is a key idea in dynamic programming. And the key assumption you're making, if you think about it, is when you solve this subproblem here, I'm sorry, when you solve this addition over here to how you add on to the match, you're not disturbing the solution that you made internally. And so that's the assumption you're making, that when you add on to the alignment, you're not going to affect the alignment inside. And that's actually true in sequence alignment. And, we can talk later about sometimes where that assumption is violated, okay? So now I'm going to add another sort of say bell and whistle to the whole thing. So we have the notion that we've talked about of matching to, now in this context, it's amino acids. They could be nucleotides. It could be any type of thing we're matching. But we have the notion of matching, say, 2 amino acids. And we have the notion that we match them, we get a sort of a goodness number. We've said that's 1, and that's in contrast to our gap penalty of a half. But we could say, well, it's really actually better, say, when you're matching, to match some amino acids which are more similar to each other, say leucine with leucine, than say matching a leucine with a charged residue. Let me say a little more on that. So obviously matching leucine to leucine, you're getting an exact match, but you could match, if you're going to make a mismatch between, say, leucine and another amino acid, say aspartic acid, which is this D, or valine, you might have the intuition of, well, it's actually much better to match the leucine with the valine because they're very similar chemically, right? Whereas leucine with aspartic acid, these are radically different. This is a hydrophobic amino acid. This is a charged amino acid. So it might not be really the best. It doesn't really match our intuitions to treat these equally. And so you could imagine creating some sort of scoring system where you have 2 amino acids and you're matching, for instance, 2 amino acids, and you're going to give just that match of 2 amino acids or that substitution a particular score. And for instance, let's start with leucine to leucine, you get a score of 1, but maybe you would say leucine to valine, you get half of that, whereas leucine to aspartic acid, you get 0, okay? And so this, of course, is an actual substitution matrix that people build up. And you can see there's all sorts of different ones. Now, this one is, I think this is a log odds one. So there's negative numbers you can get if you're less than 1 and so forth. But you can see some things are very unfavorable. Here's the L to D. And some things, let's find one that's less, let's see, D to E, you see, is a much better match and so forth. And there's, of course, a whole bunch of these matrices that people have built up. I mean, over the years, one of them is called the PAM matrix, another is called the BLOSSOM matrix, the Gonnet matrix, whatever, lots of different matrices. And where do these matrices come from? Well, there's a lot of different ways you can imagine coming up with the number in the matrix, including just your raw intuition about the similarity of the different letters. But a slightly more rigorous way of doing that would be to imagine you had some alignments of protein sequences, perhaps from looking at the protein structures. And then, and these were true. They somehow created like a gold standard of what is a correct alignment. You could imagine looking at that alignment and kind of calculating how many of a given type of substitution you have. And if you have more of a given type of substitution than not, it would maybe be more favorable. So here's what you might do. You might have these gold standard alignments, and you would calculate, oh, how many times in these gold standards do I see an A being substituted by a V, or an A substituted by an S, or a V substituted by an S, and so forth. And then you could take this frequency and basically use that, or take the frequencies of the substitutions and then use that as a score. Now, a little bit more complicated because you probably want to take into account that amino acids occur with differential frequency. So obviously alanine occurs much more often than tryptophan. So it's probably more likely that you're going to have AA pairs or A with other thing pairs in the thing. So you might want to describe the number of observed substitutions relative to how many you expect based on the frequencies. And so this would be a traditional matrix that you would construct. Here you have your substitution matrix entry as the log of these two frequencies. And then if you did this, you would basically find that, positive things are being substituted more likely than expected, obviously negative less likely, and that's what you get for your matrix. And you would simply add up those scores. Now, there's an additional little tricky bit of this too, is that the sequences in that big alignment, you imagine some are almost the same, but some are really different. And so you'd imagine let's just be very concrete. When they first did this, they maybe took all the sequences of the globins. These were a particular type of gene that people worked on a lot initially. And they took all these sequences together, and they had some that were, say, all from primates or all from mammals. But then they had a few that were very, very divergent from very different organisms, like a fish or something like that. And you would obviously see many more differences if you took the very divergent sequence. So, and actually, if you restricted your alignment to just very similar sequences, you would maybe observe somewhat different substitution patterns than if you took more divergent sequences. So there's this notion of building matrices that are based on gold standard alignments, where the sequences in the alignments really came from evolutionary distinct differences. So for instance, if you had your sequences confined to, say, just mammals or things that were fairly close together, maybe within the last 100 million years, you would make one type of matrix. But if you allowed your matrix to have an alignment that was more distant, that maybe went, look at all of metazoa, or look at all of, I don't know, maybe all eukaryotes and so forth, you would you would potentially be joining things together farther back in time, say 200 million years, 300 million years ago, and you have a different matrix. And so one of the first people to look at this particular problem sort of coined this PAM terminology, point accepted mutation terminology. And you'd have, for instance, PAM 100 to describe a matrix for fairly similar things, PAM 200 for farther away, and so forth. And then what you could do with these matrices, which is kind of fun, is you could make up a lot of the matrices. So here's actually the PAM 250 matrix. Here's the PAM 70 matrix. That's a very close matrix. And then you can look at the matrix elements of the matrix. So here you can actually look at them, okay? And for instance, you'll see that they'll be different. So let's just take an example. If you look at, well, let's see if we can, yeah, well, you can just see here. Here's the matching ileusine to cysteine. You see it gets a -2 here, and it gets a -4 here. And then what you could imagine doing is taking any of these matrix elements, for instance, let's say leucine cysteine, or leucine serine, and so forth, and you could graph how that element changes over different levels of evolutionary distance. So obviously, you would have you take this number here for the 250, this number for the 7, you can make up more matrices. And then if you did that, you'd find that some of the matrix elements kind of change in a characteristic way. And I think it's actually kind of pretty how they change. What you'd find is that the nearby PAM matrices, say the 70, the things that get the high score, the similarity is really dominated by the genetic code. So, things that you would have to only make, the genetic code is, remember, 3 nucleotides for 1 amino acid. So amino acids that were linked by only a single substitution in the genetic code would be closer together, would get a higher score, because only required 1 mutation in nucleotide-wise. And if you required 2, they'd be farther. Whereas when you looked at a farther away PAM, then the similarity was really dominated by your chemical intuition, because you can make a lot of mutations, and you would be really grouping things together that were really fundamentally chemically similar, isoleucine and valine, aspartic acid with glutamic acid, and so forth. Oops. There we go. So now I'm going to give you another bell and whistle here. So now one of the things that we've talked about is this notion of matching the entire sequence. So we remember we had when we first talked about, we had the notion of end gaps. We would, maybe we would really, try to match the whole sequence. But maybe if we're matching a sequence, there's only a small part of the sequence between 2 sequences that match. And this would be, if you think about an example, maybe we had that sequence analysis is fun and fun sequences are fun. Maybe we had another phrase that we're matching, like fun is beautiful. So the only word we're matching here is this word fun, but we want to find that small thing optimally. In a biological context, that would be like finding a motif, a small sequence motif that overlapped in the sea of a bigger sequence. And so this is a slightly different, but again, a very related problem. And so people figured out how to solve this, and they called the previous problem I solved global alignment, because you're finding the global overall alignment of 2 strings, whereas they talked about the solution to this particular problem is local alignment. And so one of the neat things they were able to do, and this is due to Smith and Waterman, who, as opposed to Needleman and Wunsch, who developed this. And this is now probably 10 years later. This is like the 1980s. They developed this approach. And so what they did here was they made three modifications to the overall procedure I described, and it's kind of neat. So first thing is they require that the scoring system uses negative values for mismatches. So it can't be just like zero. You have to use negative values. And the reason they want to do that is they do not want to allow the matrix that that you're kind of compiling, that overall summation matrix, to ever have a score beneath zero. So if you were to add on the, if you were adding in a situation where you're adding on lots of mismatches and they were negative, you would just floor them and you would say, the best I can do is zero. So you would never go below zero, okay? And then when you came to finding the best match, you wouldn't look up top at the end of the thing, but you would actually look anywhere in the matrix. And that way you could find a good match in the matrix, which maybe would actually degrade if you kept trying to align things. So they made these modifications, and that's sort of why they made them. And then they were able to find little overlapping patterns like this STVV that overlaps between these two sequences. And here's a picture now that I've contrasted the global Needham-Wunsch versus the local Smith-Waterman alignment. This is from Stephen Altschul's book. And so you can see here, it's a little bit different what's kind of going on here. You basically have the same sequences, right, that we're aligning, but because we're able to get the best score, let me see where we do it. I think here in this one over here, we're able to just focus on this little inner motif and so forth. So you get a match score of 4 with one mismatch and so forth. And whereas this one would get a considerably worse match. So it's just the difference between global and local alignment.